Translators, welcome to Learn Extra Live. Finally, we are on grade 12 Life Sciences with Aslam. How are you, Aslam?
Great, Katlik, how are you?
I'm great, thank you. Today we're doing revision. Please tell us
Starting
more.
with revision, paper
Okay.
1, the bulk of the marks, DNA, RNA, protein
Yes.
synthesis.
Yes, that's the important stuff, isn't it?
That's the stuff.
Thank you. So as you heard, guys, revision and your exams are right now, basically. I mean, you guys are already so involved in them that you need all the revision that you can get. So why not call up everyone in your class? Facebook them, SMS them, BBM them, pep text them, whatever means that mix it, whatever things that you guys use these days, tell them to join today because this is a very important show for you to pass. Facebook.com/Learn Extra is where we are at today, and we're also on Twitter. Remember, don't inbox us, write it right on the page so that we can see exactly what you say, quick like that, so that we can get back to you as soon as you can. I know that there'll be a lot of questions today, so ask them clearly and concisely so that I can shoot them over to Aslam and he can answer them directly for you. Over to you.
Thanks, Katlejo. Welcome, Mindsetters. We're going to be getting right into DNA, RNA, and protein synthesis. Please don't forget to post your questions on the page. We want your questions because that's what makes this so exciting, to answer your questions. We have our questions, we're going to go through them, but we want your questions so we can clear those fuzzy areas in your mind. Just a quick overview of what we need to do. In this section, guys, you need to know, this is DNA and RNA, you need to know the structure and functions of the nucleus. Now, guys, that is grade 10 work. Just what the nucleus is made up of, what does it do, and more specifically, how is it involved with these structures, DNA and RNA especially. Then we go straight into DNA. We want to know about DNA. We want to know the structure of the DNA. We want to know how it works and its function, and how it makes copies of itself, replication. We then move into the RNA structure. When we look at two different structures in life science, generally, we would always then also require a comparison. We must be able to compare the structure of RNA to that of DNA. We then move on to the process of protein synthesis, which is made up of, as you know, two important processes, that is transcription and translation. We then move on to gene mutation. We need to know what mutation means, definition of a gene mutation. We must also know the types of mutation and the effects of these mutations on protein synthesis. In other words, when a mutation takes place, first of all, what do we mean by this word? Secondly, when it does take place, what are the different types? And finally, how does this affect protein synthesis? We then move on to DNA sequences, or some books may call it DNA profiling or DNA fingerprinting. That's the first part. And also how DNA sequencing can help us to see the relationship between different organisms. For example, how closely related are we to a pumpkin? Katleho, you think we're related to a pumpkin?
I don't think so. I like to think we're not.
Well, we can find out using DNA sequencing how closely we are
And
related
are we
to a
close?
pumpkin. No, we're not, fortunately.
Unfortunately.
Katleho. Fortunately, we're not.
Good.
But we are closely related to the chimpanzee.
Yes, I know that one.
Okay. Lastly, we look at recombinant DNA technology. That word gives me problems all the time. Recombinant DNA technology. In other words, how we can use our knowledge of DNA to advance medicine and our knowledge of organisms generally in terms of the technology, how we can cure, how we can study diseases, etc., using DNA. In other words, how we can produce medicine also using DNA. Interesting stuff, guys. This section should keep your juices flowing all the time. And today, in today's time, these two sections here, with all the lovely programs on TV, all the detective series that you're watching, these things crop up over and over again. DNA fingerprinting, how they find criminals, etc., how they find cures to outbreaks of various diseases. All these things are all that stuff that you are seeing. The time when we wrote this stuff, we didn't do all of this anyway, but, Katlego, in our time, there was no medical series like this.
Really?
That's right.
Wow. So you guys had no CSI and
Nothing.
Oh my word.
I remember we had one lawyer program that was, and one was a university
and order,
pastor professor. No, before law. I'm coming before law and order. Look at the color of my beard, Katlego.
So do you guys have Rescue 911?
Before that. even.
Before that even. Oh, wow.
There we go. All right, guys. That's the overview of what we have to do. Now, let's go straight into the question. Remember, we have taught all of this before. It's on the on the Mindset web page, etc. You can look at that. You can look at our notes, etc. Now, we have selected some new, fresh questions to test all of those topics that we have spoken about. So we're looking at the first question here. It says the diagram below shows part of a DNA molecule. So they're telling you up front that this is a DNA molecule. Some questions may not be so explicit. They may say the diagram below shows a molecule. Or they can say it shows a strand of a particular nucleic acid. Remember, we only have to concentrate on two nucleic acids, and that is DNA and RNA. Let's go into this question. So they've given you a lovely diagram there, and they say provide labels for parts 1 and 2. So obviously, guys, you need to be able to label messenger RNA, transfer RNA, DNA, etc. That's the first level of your knowledge. You need to be able to know the structure. And the best way to learn the structure, label a diagram or draw the diagram. Draw the same diagram and label it. So in that way you are learning. Don't learn a chunk from a paragraph or a chunk from a textbook explaining the structure of DNA, explaining the structure of RNA. Get the picture in your mind's eye. And that way, when you are explaining it, you are closing your eyes, you're seeing the DNA, and you are explaining on the paper what you see. Remember that. So labels for 1 and 2, where's 1 there and 2 there. The one on the outside is the phosphate. Not phosphorus, it will be marked incorrect. Phosphate. And the one on the inside, because they said DNA, they would require you to be specific. Last year in the exam, a similar question came up, and the learners wrote sugar. And this was marked incorrect. Why? Because it was a DNA, you had to say, the examiners say, you must be able to say, if you are using DNA, that this is deoxyribose sugar. You must be specific, guys. And in RNA, that would be ribose sugar. Deoxyribose sugar in DNA and ribose sugar in RNA. We're touching on both as we're going along, and we'll come to some of those questions again later. Good. Still with the same question, 1.2 says, during a particular process, the DNA molecule presented in section B of the diagram, this one here, is built up from the molecule shown in section A. This is also part of the DNA molecule, and they're saying in a particular process, this part is built up from here. They wouldn't say that, they wouldn't use the word copied in this question, because if they use the word copying, it would have given the answer. So they use the word built up, but it's the same process. And the process that we are talking here, process is called replication. DNA replication. Rather, write the word DNA replication so you get the full mark there. In what part of the cell does this process occur? Yes, you got it right. That is in the nucleus. If you want to be more specific, you can say in the nucleoplasm. But nucleus is correct there. Identify parts 3, 4, and 5. Go back to the diagram. 3, 4, and 5. And if you look at them, 3 is adjacent to adenine, 4 to thymine, and 5 to cytosine. And if you remember our lesson on this section, I told you to remember at, and I also asked you to remember cute girl. Katlecha we're talking about now. Cute girl. Cute lady.
Really? Lady. Not cute. I don't like cute. Maybe something else. I'll think about
we'll
what
think
else.
about it. You'll let me know after that. Okay. So we got adenine at. So with adenine, 3 would be thymine. I'm just going to put the letters. You know what they stand for. Thymine. And if adenine combines with thymine, then what would thymine combine with? Obvious. Adenine. AT, at. And obviously the last one, C, cute, will be guanine, number 5. So what the examiner wants to know here is whether you understand how the nitrogenous bases combine. In other words, which one combines with which one. So you need to know that as well. At, cute girl. Remember that? You safe. Remember in RNA, this changes a little bit because this guy is not present in RNA. But we talk about that a little later. Okay, we've identified those parts. We're still with the same question. During a particular process, no, that's the same one there. Okay, yeah, we kept the question, sorry. Name the chemical bond that holds the 2 threads of section A and B together. We're talking of the same process. They want to know when this thing is done, when we make this copy, how are these things held together? How are they held together? And they held together by hydrogen bonds. And it would be useful to remember when you're saying it, weak hydrogen bonds. Although in a memo, this would be in brackets, and we would give you the mark for hydrogen bonds. But you keep that in mind because we're going to ask you something about that later, and then you'll be able to say that. If only 2 labels of section B are changed, this will represent an RNA molecule. Provide the numbers and the new labels. Go back. We come here. If we change 2 only in this particular diagram, remember, we must go back to what we said. This would have been thymine, this would have been adenine, this would have been guanine. And remember what we labeled this as, deoxyribose sugar. They want to know if we change 2 labels in this diagram, this molecule would not be DNA, but RNA. Which 2 are they? Number, they're saying, provide the numbers and the new labels. So there's 2 parts to the question. Go back to the diagram again. Obviously, I've already given you part of that answer, or the whole answer earlier in my explanation. And the 2 would be number 3 and number 2. Number 2 and number 3. In this case, number 2, the label would be ribose sugar. And number 3, instead of thymine, you have uracil. So that is our answer there. Remember, look at the mark allocation. 4 marks given here. So how are the marks allocated? 1 mark for giving the number each, and 1 mark for giving the new label to make this DNA strand into an RNA strand. Excellent stuff, guys. We need to know how to answer that. Next question says, the diagram shows that whatever during a particular process in this, we're carrying on with this thing here. If section A remains a DNA molecule, what kind of RNA molecule is represented by the newly labeled section B? Remember what now you got to think carefully. Obviously, you won't have the question all on 1 page, but you got to think carefully. What did you do the step before? You made that DNA part, you made it into RNA. And you know that we have 3 types of RNA. Messenger RNA, transfer RNA, and ribosomal RNA. They want to know from you if this new RNA molecule was made from a DNA molecule. then which of these three is this RNA? And you needed to know your protein synthesis for this, obviously, guys. And the answer, obviously, is messenger RNA. Because DNA gives rise to messenger RNA, and messenger RNA, in turn, codes for the specific amino acids to be carried by the transfer RNA. We're going to go into protein synthesis in detail later. We'll talk about it again. But for now, just so that you are with me on the same page in terms of what I'm saying here. That's why the answer here is messenger RNA. The last question here, G, says tabulate 3 structural differences. Structural differences. You can't talk about the one being in the nucleus and the one being in the cytoplasm, because that is not a structural difference. We are only interested in structural differences here. And obviously, I've given you some already. DNA, before we even, all right, let's go to what we've spoken about. DNA has adenine, guanine, cytosine, and thymine, whereas RNA has adenine, guanine, cytosine, and uracil. Secondly, I said DNA has deoxyribose sugar, and RNA has ribose sugar. The last difference comes in the fact that DNA is a double strand, and RNA is a single strand. Some textbooks and some authors talk about the fact that in DNA you are not going to get hydrogen bonds. In DNA you'll get hydrogen bonds, and in RNA you do not get hydrogen bonds. But in a memo situation, that mark would be added most of the time to this one here. Because, and that is why it doesn't have a hydrogen bond. Because DNA is double-stranded, it has a hydrogen bond, and RNA will not have hydrogen bonds because they are single-stranded. So that would be an alternative to this particular one. So in other words, if you gave both, you may sometimes, depending on how the examiner puts out the memo, you may not get both marks. So please be careful. These are the 3 major differences between DNA and RNA in terms of structure. If you want to go further, if they just kept out the word structure there, then you're going to talk about the different types of RNA and the different types of DNA. Remember, we get nuclear DNA and we get mitochondrial DNA as well. And we learn a lot about mitochondrial DNA in a section on evolution. So you could have used that. Then you could have used another difference about where DNA occurs and where RNA occurs. You could have done that as well. So those are some of the other differences if they did not specify structural differences. In that sense, remember that. Also here, let's look at the mark allocation. You would have got 1 mark there, 1 mark there, 1 mark there, 1 mark there, 1 there, 1 there. That's 6, plus 1 for drawing the table because they asked you to tabulate the differences. Also in a table, keep the differences adjacent to each other. You can't talk about thymine here and next to it ribose. Although it's correct, but it's not a difference here. So you lose the second mark. So keep those things in mind. Remember, tabulate, put your table, complete the columns and the rows, and keep your differences together. With that, I've said a lot.
Thank you. We're going to go for an ad break. I know that you guys need a bit of a refresher, but as soon as you refresh, get back on that seat and watch us again for the next segment. See you right now. Welcome back, guys. If you have just joined us, we are doing life sciences for grade 12, and today we're working on paper 1 revision. It's already been very nice, and you guys are already enjoying it because it is very relevant and important to what you are dealing with in your lives at this very point in time. So in case there is something that has been bugging you or something that will help you for the future in your exams or was bugging you for the last exam, whatever it may be, and it's related to this that we're doing right now, please don't fear to send us questions because that is exactly what we're here for. Over to you, Aslam.
Thank you, Katlego. Let's move on, guys. Welcome back. I'm hoping you revitalize. We've done a little question on DNA replication, and the differences between DNA and RNA. Now we're moving on to a question on protein synthesis. The question says, the diagram below shows one stage in the synthesis of a protein at a ribosome. One stage in the synthesis of a protein, so we're dealing with protein synthesis at the ribosome. And that is an important clue. And I'm hoping you're watching how I underline the key words as we're going along. That is the most important thing, guys. Most of you know the answers of questions, but you don't know which answer fits where because you do not read properly. And once you get into this habit of underlining the key words, your job, half of it, is already done. You just need to then put those answers together. So they've given us a diagram here. They're showing us there something's happening there on top there, a transfer RNA. That's another clue in the diagram. Another clue is given is amino acids. When you see the instruction, the little introductory paragraph of sentence and the diagram are giving you clues to the rest of the question. And they're talking about some bond there. They're giving you some nitrogenous bases. They tell you this is messenger RNA there, and so on. They say, name this stage in protein synthesis. What is the name of this stage? And obviously, like I said, the clues were given to you. The fact that they mention ribosome, the fact that they're talking of transfer RNA, the fact that they're talking about amino acids, that all leads you to the answer that this is translation. Remember, when we started, we spoke about two stages in protein synthesis. The one was transcription, and the other one was translation. That's what we're talking about here now. Remember, transcription takes place in the nucleus, and translation takes place in the cytoplasm, more specifically at the ribosome. Just by the way, the word transcription means to make a copy. Transcribing, if your teacher wants to use high terms to you when you are looking at your friend's work, or if he notices that your work and your friend's work are the same when he's marking your assignment, he says, Katlejo, why have you transcribed from Jonathan? What does he mean? You've copied. All right? So transcription means you've made a copy. Remember that. So which copy are we talking about? From DNA. And what do we form? Messenger RNA. Those are the most important things you know about transcription, and you need to explain that. We'll worry about that when we deal with the essay question. Transcription and translation, translation. If I start talking to you in Arabic now, or Gujarati, or Urdu, or Swahili, or any other language that you are not familiar with, you would need somebody to translate that to you. So in other words, we're taking something and we are translating it, or we are changing it to a more, or to a different structure in this case here. So we've taken a code that we got from the nucleus, and we're converting that code into something else that we can use, and that is called a protein, and the process is called translation. Name the bond X. Where is the bond X? Not James Bond, the bond X. Maybe we must come out with our own movie, Bond X. But here we're talking about, and what are these things? They gave you a label here. These things, 1, 2, 3, 4, 5, are amino acids, or amino acids, whichever you prefer, as long as you spell it correctly. There's a bond between two amino acids. And that bond, your grade 10 work would have informed you about this, but in grade 12, we touch again on that. That is called, and let's go back there, down, and we say that's a peptide bond. So a peptide bond forms between two amino acids, adjacent amino acids. Two amino acids together, in this case here, would be called a dipeptide. The third one is also there, so this is a tripeptide. Two amino acids, dipeptide, 3 become tripeptide, and then on we call them polypeptides. What type of bonds are indicated by the dashed lines, and state what characteristic do they have that makes them suitable between the nitrogenous bases? What type of bonds are indicated by the dashed lines, these ones here? And what special characteristic do they have to make them suitable as bonds between nitrogenous bases? And remember when I asked you just now, what are those bonds between two adjacent strands? We said they are hydrogen bonds, and I specifically said we must add the word weak in front of them. Now there's a question that's addressing that. What type of bonds? We say they are hydrogen bonds. And they said, what characteristic do they have that makes them? They are weak bonds. That's the answer to that one. Katlego, we had a question earlier about this.
Yes, we did. Let me get it for you. It's so down the page now.
What
so
would happen
you
if...
something about the strong
The
bonds.
question was, yes, good. The question was, what would happen if the the hydrogen bond was strong?
Okay, here you go. It's the question by Roxanne.
Yeah.
She asks, what would be the consequence if the hydrogen bond was strong? That's it. You got it right.
Okay, so she wants to know, Roxanne wants to know, what would happen if these hydrogen bonds were strong? We said that the weakness makes them suitable to form between the nitrogen space. Now we need to go back to our first question, which dealt with DNA replication and transcription. And now we're looking at translation, where the transfer RNA molecules combine with the messenger RNA. Look at this transfer RNA here. It has combined and it moves away. So it means that these bonds that form must be able to break easily. If they were strong bonds, then the bond would not break and the strands would not be able to separate. And if the strands could not separate, DNA would not, Katlego, be able to make a copy of itself because it cannot separate. Messenger RNA, once it formed on DNA, would not be able to detach, and therefore it would not leave the nucleus.
Okay.
And therefore it would not get here, where it is here down. And thirdly, guys, the transfer RNA, once it combines with the messenger RNA, would not also be able to detach. So it's stuck there, and consider the transfer RNA as your taxi. If your taxi gets to the spot to drop off passengers, Katlego, and it has a puncture, which is famous excuse I get every single morning. We had a puncture. The taxi broke down every morning. So if he gets stuck there, he can't come back to fetch more passengers. And in this case, the passengers are amino acids. So in other words, if transfer RNA gets stuck here, we cannot get more amino acids, so the process of protein synthesis in all these steps stops. So the ultimate, the end product is that protein synthesis will not take place. But now, I give you this scenario as well. It's all very well to say in this type of a question that Roxette has posted that protein synthesis will not take place. You will get 1 mark for that because that's the last statement that you have to do that. But if I have a heart attack, I will die. I may survive, but you can give us, I may die as well. So I'm going to die right at the end. But what has happened between the fact that I got a heart attack till the point I died? Why did I die? So in other words, we need to explain the steps
before
to get
that.
There. Okay? So what are those steps? Replication will not take place. And if replication doesn't take place, then the DNA doesn't split and therefore it's not exposed to the messenger.
Yes.
Secondly, messenger RNA, if it does perhaps connect with one DNA strand, it cannot separate. So if it doesn't separate, it cannot then go out and it cannot give the code to transfer RNA. Thirdly, if transfer RNA gets stuck to messenger RNA, it cannot separate, it cannot fetch more amino acids, so the process stops. As simple as that. So in other words, in this question, where they say, that's why I didn't explain this because I wanted to deal with that question, what characteristic do they have? The characteristic is that they are weak bonds. You would have got the mark here anyway because they only ask you what characteristic. They have weak bonds, and if they said explain why, so that they can easily break during replication, transcription, translation. In all 3 processes, we need this bond to form and to break easily. I'm hoping we're on the same page. Give the messenger RNA codon that would code for the transfer RNA bonded to amino acid number 5. We go back. Amino acid number 5. That's amino acid number 5. It's on this transfer RNA. There's the amino acid here. Here's the transfer RNA under, and the letters given there are GUG. Go back to our question there. So our transfer RNA is GUG. They want to know, if this is the transfer RNA, which part of the messenger RNA would this particular amino acid or this transfer RNA combine with? Obviously, you know cute girl, so the girl is cute now. G with C. And obviously, if that is G and C, so that will be G and C. This one is always the tricky one. If they tell you U here, it's not as tricky when they say U. I'll explain just now as well about that. What happens on this side, if it's U on this side, then we get A on that side, because the equivalent to U in a DNA is thymine. And what combines with thymine? Adenine. So A combines with U. A, U. Remember that. Okay? So A combines with U there in that case there. Remember that this is the messenger RNA. They can then ask you after that, so what DNA strand gave rise to this one? So now you're going right back to the nucleus again. How did this one form? Again, if that's the case, that will be G again. That would be G again, obviously. And here now, because this is DNA, this would be T. So what's this? I don't want to change that to show you something. I don't know why I'm coloring there, but anyway, look at these two. That one and that one. The transfer RNA and the DNA are the same, excepting for the transfer RNA does not have T. So in place of T, you can't put U there. I mean, you can't put A there. That's where the confusion comes. Many people, in this case, they'll put A there. It's not A there, it's U. The A goes here by messenger RNA. Consider the messenger RNA to be in the middle. And the messenger RNA is opposite of the DNA and opposite of the transfer RNA. And that's how we get our answer to a question like this. I'm hoping we understand this, because this one here is easy jam marks. Moving on. A mutation of the first messenger RNA codon occurred, and it read CGU instead of CCU. Katleho? You remember that one? About those
Yeah.
mutations
Yes,
and how your name
yes.
got mutated.
Yes, I remember that.
Remember our first encounter,
Yes.
right?
Yeah.
Pat and cat.
Pat and cat.
There
I
we go.
remember
All right.
that.
So this is a similar situation. One letter has changed. It read CGU instead of CCU. So one particular nitrogenous base has been replaced by another nitrogenous base. Nothing has been added and nothing has been deleted. Just one nitrogenous base has been replaced. They want to know, would this be a point or frame shift mutation? Obviously, in this case, it would be a point, because only one nitrogenous base was replaced there. Explain the differences between the two types of mutations mentioned. And we are going to deal with it again in the essay, but let's just deal with it now. Then when I get to the essay, I'll refer you to this as well. So the one is obviously point, and the other one is frame shift. Here, only one nitrogenous base is replaced by another. Okay? That's point. Frame shift, if we add or delete a nitrogenous base. Now the famous one they use there, the dog ate the cat. The dog ate the cat. If we remove this one here, then what happens, remember, we must have base triplets. Everything moves. That becomes that becomes whatever they add to that there in the end. Now, can we read the second sentence? No. No real words are found in the second sentence. And what has happened? There has been a shift. The whole frame has shifted, left in this case. Okay? The whole frame has shifted left. So this is a frame shift mutation. When one is, in this case, deleted, or let's see what happens if we add. If we had the dog and we add a G there, now it becomes that. So in both cases, in this case, obviously you can read the dog, up to the dog. After that, unless you switch to Afrikaans, then you're going to say the dog gat. Okay? Or if you want to use your colloquial terms, gat for gun or whatever. But English, if we're using one language, then obviously we cannot complete the sentence. And that becomes complicated for us. And that's where the problem is. In the next question or in the essay, we're going to look at what are the implications of this in nature. The next question, so we've completed that one. And this type of question is linked to what we've just done. So we look at this question as well. The diagram below shows the sequence of nitrogenous base of a strand of DNA. So they're telling you it's DNA, which codes for a part of an insulin protein molecule. AAA, CAA, TAC. They want to know from you, write down the messenger RNA. Insulin, by the way, is a hormone. And one of the characteristics, paper 2 now, guys, of hormones is that most hormones are protein. Insulin is one such hormone that lowers the blood sugar level, just so that we keep that in our head. Don't see this and now start talking about sugar levels and the rest of it, because we are not talking about the functions of insulin here. All they want to know from you here is that if this is the DNA that codes for the insulin, then what is the messenger RNA going to be for this one? If that's AAA, now here, it should have been at TTT, but because it's messenger RNA, that will be U. Left to right. You've got to write it in this order. U, C would be G, A would be U, T now doesn't become U. The opposite of T in this case is A. The opposite of A is U and G. So you write it in this way. U, G, A, G, and you get your 3 marks that way. With that, back to you, Carlejo.
Thank you. So guys, we are going to go for an ad break. I know I can see all the questions on the page, and I am going to show them to Aslam. I know that some of your questions have already been answered. That's why you haven't received a reply from me. So just stay focused and remember not to move from your seats, because we will be right back. Welcome back, Mindsetters. We are doing life sciences, and it has been absolutely awesome because it is revision, and revision is always good. I mean, always good, because you can never know too much, really. If you do want to pass those exams, especially for those of you who want to get exemptions, this is a very important show for you, and I'm glad that you guys have joined us and you've been absolutely focused, because the questions are absolutely amazing as well. Aslam, there's an interesting one.
Yeah.
Mutlati says a good one is good couples always together, for GCAT.
Good.
That is awesome, hey?
Good couples
always together. There
you go, guys. You can make your own mnemonics. TACG. Good couples always together. There, I've learned something great today, and I'm going to definitely. What's the name again? Mutlatsi. Thank you very much. I'm going to be using that. I hope you haven't patented it yet, because I'm going to be using this one. Good couples always together. There's your G with the C, A, and T. Great, guys. In this way, you make the mnemonic that makes sense to you. Whatever makes sense to you. We're giving you guidelines, so great for that one. Any other questions there?
Yes, we do have questions. We have one by Sfiso. He asks, what's the difference between monomers and nucleotides?
All right, let's look at these 2 words, monomers and nucleotides. Sfiso, a monomer is a building block, much like a brick in a wall. The brick is a monomer. The single smallest unit that a wall is made up of is the brick, and the brick becomes the monomer. In DNA and RNA, the nucleotides are monomers. These are not synonyms. They're not synonyms. A monomer is a building block, but like the brick was a monomer in a wall, the nucleotide is a monomer of a nucleic acid. A nucleotide, by the way, is made up of a phosphate, a sugar, and a nitrogenous base. For example, in this case here, this is an adenine nucleotide. And it's a building block of DNA. So therefore, it's a monomer as well. So the word monomer means building block. A nucleotide is made up of phosphate, sugar, and nitrogenous base, and a nucleotide is actually the smallest part of a DNA, and those are the monomers of a DNA.
Okay.
One more question
Yes,
there?
there was one more by Vuyelwa. Vuyelwa asks, what are the differences between lethal, neutral, and fixed mutations? So that's lethal, Vuyelwa.
Vuyelwa, lethal.
Oh, sorry, Vuyelo.
Vuyello. Okay,
yes.
good. Vuyello wants to know about lethal, fixed, and neutral. Lethal, the lethal weapon. What's the guy's name again? Mel Gibson.
Yes.
Lethal weapon. Lethal weapon, a weapon that can kill you. A lethal mutation is a harmful mutation that leads to the death of an organism. That means it's so bad that it's going to kill you. That means you cannot survive with that mutation. Straightforward. We go from there right to neutral. Neutral, guys. If I tell you I'm staying neutral in this matter, or if the car is not in drive or park or first, second, third, fourth, fifth, or reverse, it's in neutral, it means it's in no gear. So neutral means it does not have no effect actually I should say, no effect on the organism. It is there, but it's not going to make a difference to the organism. And a fixed mutation is generally one that becomes beneficial. That this mutation is so good that it gives certain organisms advantages over other organisms. And therefore, these organisms survive, and the ones without this mutation, they die. So this becomes a beneficial one, and it stays with the organism. So it's a fixed mutation. Simple explanation, guys. Don't look into too deep into this. Just remember this what I have explained here. That's all for now?
That's all for now.
Great, guys. Great. Keep bringing those questions in. Now we are moving on. We've covered a bit of protein synthesis. We've covered our DNA and DNA replication. We've covered some of the mutations. We're moving on to technology now. Gene is a segment of DNA with bases in a certain sequence. Human genes can be isolated and then inserted into bacteria. The figure below shows how insulin can be synthesized by inserting the human insulin gene into a bacterium. After this has been done, the bacterium is known as a transformed bacterium or a transgenic. Transgenic because we have taken a gene from one organism and put into another organism. This is just one example of that. So they're giving you the whole story here. By the way, this one here we've taken from one of the high schools in the Western Cape, and we've adapted it for ourselves here. Great question. Normally we get a diagram that's fixed here. They've made their own diagram. There's a human cell. Here they've taken the chromosome from the pancreas of this human, have isolated it. They have cut that insulin gene. At the same time, they get a bacteria. They take a plasmid. They isolate it. They've taken a plasmid, and they've isolated this plasmid, and they've cut it. Look at this diagram here. Look at this diagram there. They have cut it using an enzyme. These enzymes are called restriction enzymes. Restriction enzyme. By the way, this enzyme works like a scissor, not a physical scissor, but a chemical scissor that breaks this plasmid and leaves a spot there. Now what they do, notice what they've done here. They've isolated the DNA. They've taken out the insulin gene. They've cut it, also the same process, restriction enzymes. They've taken this enzyme, this gene, and inserted it here, also with an enzyme. And they then take this whole plasmid and put it back into the bacteria. Now what do we have? We have a transformed plasmid into this bacteria, so we have a transformed bacterium. They allow the bacteria then to reproduce. And these bacteria produce more and more of the same. They then remove the insulin that is formed, and they store this insulin. And this insulin then is available to diabetes sufferers in an injection form or in a vial. a little tube where they can inject themselves. The first question here is, what is a plasmid? And obviously the diagram is telling you that. What is a plasmid? It is a DNA, so it's a ring of DNA found in the bacteria. What molecules is the insulin gene made up of? What is it made up of? And in this case here, obviously these two questions wouldn't come the same in an exam situation. I've taken it from different papers. And we answered that a little earlier, that insulin is made up of protein. The last question gave you that. Okay? What would we call the four identical cells at the bottom of the diagram? These four cells that came from one mother cell, they are identical copies. And if we use this process to make identical copies of organisms, we call the resultant organism a clone. Clone is a copy of a cell, or of an organism for that matter. We're carrying on with the same question. Suggest what type of reproduction occurs in the transformed bacterium. Now, they do not expect you to know the specific type. And if you do reproduction, you know you learn two types of reproduction, asexual and sexual. They want to know which of these two would it be. And you would say asexual. By the way, just for our knowledge, the actual type of reproduction, the example of asexual reproduction that takes place in a bacteria is known as binary fission, where one cell, by mitosis, makes many cells. So the one cell divides and the chromosomes go to opposite cell. The cell pinches in the middle, we get 2, 2 make 4, 4 make 8, and so the story goes. But it's asexual. Suggests why this type of reproduction is needed for the large-scale production of insulin. We want insulin. What insulin? The insulin that is found in humans. We want to use that insulin. Can sexual reproduction take place here? No, because when sexual reproduction takes place, a variety is introduced. So why do we need this? Because we want identical copies of the insulin. We do not want a variety here. In this case, we do not want variety. In this case, variety is no good. We don't want to change it. We want it the way it is. That insulin must do the work for us. Explain two advantages of producing insulin from bacteria rather than obtaining the hormone from the pancreas of an animal. Now, in this case, they can ask this question both ways. They're asking here the advantages of doing it this way. Or they could reverse it and say, explain two advantages of obtaining the hormone from the pancreas of an animal as opposed to getting it from human insulin. Can you see how that can be done? So obviously, you've got to take this disadvantage and make an advantage, or the other way around. And what are the advantages? First of all, if we needed the pancreas of an animal, we needed to kill that animal to get it. So therefore, we're going to avoid large-scale killing of animals. Okay? But notice what they said there. Oops, sorry. Notice what they said there. They used the word explain. Why? Because you must give a reason now. Why does it do that? Because we use human insulin. Okay? The next one would be, obviously, there are fewer or no side effects. Because, again, we're not using animal insulin. And thirdly, there may be no religious objections as we're not using animals. Okay, certain religions have objections when you use products from an animal. For example, myself, I cannot use any product from an animal that hasn't been slaughtered correctly. I cannot ever use, whether slaughtered correctly or incorrectly, any animal that is, well, not any animal. I cannot use any pork, pig byproduct at all. So different religions may have different objections to that. So therefore, this way, we're not using an animal, we're using human insulin, and therefore, there's no need for religious objections. I've just given you 3 there. I'm going to have to leave this question out. We wouldn't have enough time to do that one, so we will have to do that some other time. I just want to go to this last one here, because this is just a discussion. It's not going to go into details of the answer of this question, because we've covered most of it already. I'm going to take the core, and we're going to break it down. I'm only putting this because it's an essay type of question, and we need to learn the skill. Somebody asked me on WhatsApp yesterday, one of my Star School learners asked me about the same thing. And I've explained it on WhatsApp. I'm hoping she's watching today, and maybe, not this question rather, but just about writing an essay. So they say, describe how proteins are synthesized. Explain the difference between a point and a frame shift mutation, and the possible impact of these types of mutations on the formation of proteins. So in effect, guys, you need to know how many parts there are to this question. And in this case, there are 3 types. First of all, how proteins are synthesized, 1. And what are we looking for? We're looking for the verbs. Whatever comes after the verb is what you have to do. Describe what? How proteins are synthesized. So in other words, you need to describe protein synthesis. Okay? Explain the differences between what? A point and a frame shift mutation. That's the next one. And the possible impact of these types of mutation on the formation of proteins. So there are 3 headings here that you can put in. So you start with protein synthesis. And I'm just showing you a plan. I'm not going to explain the whole thing. We do not have time. Protein synthesis will be broken up into transcription, which you will explain, and translation. Okay? From there, you will talk about the point and a frame shift, which we've already done. What's the difference between a point and a frame shift? And the last part, you will talk about if a point mutation takes place, how does this impact on the formation of proteins? And if the frame shift takes place, how would this impact? Now, generally, if you look at the point shift, you know at the point shift, look at me what I'm saying, point mutation. The point mutation affected only one nitrogenous base. It did not affect everything. If it does not affect the type of amino acids in a big way, then it may not have a serious impact. However, if it messes with an important amino acid, it can cause problems. Okay? Whereas with the frame shift mutation, just by adding or deleting one of the nitrogenous base, the whole sentence is wrong. And therefore, the DNA, if it happens to the DNA, it's going to happen to the RNA and so the protein. And with that, guys, I will leave you in the capable hands of
Thank you, Aslam. What an awesome show. Thank you for that. And thank you guys. You know, Aslam, there's a Facebook message here by Danessa. She says, Aslam is the best life science teacher ever. I understand everything he teaches. Aslam rocks. She's lying. I think she's telling the truth. Many of you feel like that, I know. That's why you all joined us today. Thank you for an awesome show, guys. From me and Aslam, thank you, guys, Mindsetters, grade 12s. You guys are writing exams soon. We wish you all the best in the world, and we know you'll do fantastically. Join us again next week for more life sciences. Bye, guys.